Declining soil fertility continues to hinder agricultural production, especially among resource-constrained smallholder farmers. There are numerous studies on different soil functions, namely water and nutrient supply, carbon sequestration, organic matter recycling, and soil biodiversity. However, the importance of oxygen levels in soil and their significant impact on different parameters of fertility have gone unnoticed. Clay formation, the thin layer of soft soil that covers the Earth's surface and have the necessary characteristics to support plants' life. But in the biogeochemical model of the Earth system, molecular oxygen is always present into clays. And probably this element constitutes a fundamental first step in the formation and organization of fertile soil in which organic carbon is formed and whose presence inside clay constitutes a univocal sign of soil fertility. Therefore, an effective form to speed up the process of silicate weathering and thereby clay formation could be the artificial oxygenation of harsh soil as very first spark of clay formation. Our project dates back to 1990, when we began an observational study on the morphology of the tiny blood vessels of the optic nerve in humans. This study lasted 12 years, until 2002, and included the ophthalmological records of 6,000 patients. But a few weeks after this descriptive study began, some details that we observed in the ocular tissues surrounding the optic nerve caught our attention, and therefore were included as variables in our working hypothesis. And the conclusion we reached after that hard work was that our body does not take oxygen from the air that surrounds us, but from the water contained inside the cells that make us up. Just as it happens in plants, our discovery demolishes the completely theoretical hypothesis that became the prevailing dogma to this day, once Lavoisier enunciated it in the 18th century. And it implies the need to rewrite all textbooks on biology and related subjects, such as medicine and agriculture, for example. Given the evidence that we have found in our experimental laboratory about the enormous importance of the presence of molecular oxygen, as well as its origin, inside the cell as well as in other processes related to life, we inferred that the continuous presence of molecular oxygen inside the soil could be a very important trigger for the formation of the various components that, together, determine soil fertility. To do this, we planned several experiments in which we included a new material that we developed to replicate the biochemical process by which the eukaryotic cell extracts molecular oxygen from the water it contains inside, and which we achieved since 2007. In the video, you can see the continuous formation of oxygen bubbles that originate inside the material and that, like the human eye, do not require electricity or added chemicals, and whose useful life is like that of a human being, which was to be expected given that it is a material that we developed based on the biology of the eye. And just as in the case of wastewater treatment, the effects of the constant infusion of oxygen gave us surprising results, so the possibility of the formation of fertile soil with the constant presence of oxygen alone was a new and exciting possibility, so that we got to work. The photographs, taken from one of our experiments, illustrate the placement of our material, QBLOCK, at the bottom of the container, then placing pumice sand, and then covering it with water. The periodic photographic control of the experiment allowed us to detect around 4 months later the first signs of the formation of organic carbon, manifested by small grains, which over the following weeks hatched more and more quickly, until it became an explosion of life. We have continued this experiment to date. Our observation that the human body does not take oxygen from the air but from the water that the cells that make up us contain inside constitutes a revolution, since it turns out that oxygen comes from the inside out, but not only in plants and in humans, also in water. When oxygen comes from the bottom up, the improvement in the various physicochemical characteristics of the water is substantial, and now we find that it is also in the soil fertilization cycle, which is not strange, since the formation of clays requires the presence of molecular oxygen. The formation of clays not only requires water, but it also constantly releases oxygen, and we achieve this by burying our new block material at a depth of about 30 centimeters. The difference between a sterile soil and a fertile soil is the continuous presence of oxygen inside, and now we can achieve this without wasting electricity by applying QBLOCK, a registered trademark, to the subsoil. Among the great advantages of our novel method is the fact that no electricity is required to generate oxygen, nor is it required to add chemicals. Furthermore, the useful life of our QBLOCK is like the life expectancy of a human being. We appreciate your attention, and our email is comagua2000@yahoo.com.
Pégale en la mesa. Pégale. ¿Y se mueve? Mira, el agua se mueve
y la bola no.
Seawater has around 150 times more CO2 than atmosphere. CO2 does not combine with water, only is solvated. Thus, seawater does not support life anymore. Nature's solution is by hydrating CO2 through the dissociation of water, as in plants. But until now it was not possible to do it in water, because it must be replicated as it has been done since the beginning of time. But now it is possible thanks to the material we developed based on the biology of the human eye. Our product QBLOCK does not require electricity or added chemicals, and its half-life is 25 years or more. Please write to comagua2000@yahoo.com. QBLOCK, the natural and ultimate solution to CO2 challenge. comagua2000@yahoo.com.